Starting. Hello everyone, it's great to see you again after the short break of last week. And Reut and I are so happy to join you again this week. And today we are going to talk about the technologies that help us discover the life in the universe. So if you have any questions during the talk, please post them in the live chat box that's around the corner, and we will answer those at the end of this session. Also, stay tuned until the end of this session because I have really amazing announcement for you guys. So without further ado, let's get started with Reut.
Hi, everybody. It's good to see you. I hope you're doing very well. Thank you for coming back to our amazing series of introduction to astrobiology. Today we have a really nice chapter talking about detection of life forms, what kind of methodology, what kind of technologies we like to do. And without further ado, welcome on board. Everybody who's new, don't worry. I always do like a short summary of what we did last time and then in the series in general. Don't worry about it. So I'm going to share my screen now. And we're going to start on. Okay. Is everything okay there?
Yes, I think it's good.
Great. Okay. So when we talk about astrobiology and the universe, so far we had very interesting topics to talk about in this introduction to astrobiology. Hello, everybody. My name is Reut Abramovich. I work at the Dead Sea Arava Science Center in Israel, in the south of Israel, in the Negev. It's a big desert out there. So far we talked about three interesting topics. We talked about the beginning of life on planet Earth. We talked about life on Earth and its evolution. We talked about Darwin, about Lamarck, about some other notions about how evolution might happen elsewhere. We talked about extremophiles and how we have different extremes in environmental condition, whether it's temperature, whether it's pH, whether it's pressure, whether it's many other things, and how we know today microorganisms are withstand these conditions. And today, with all that knowledge behind you, and if you haven't, you're welcome to go and watch those YouTubes before. Today, we're going to talk about technologies for life detection. And astrobiology, as a rule, centers about life. So after we know a lot about the diversity of life, and all these topics have been discussed before, what are the most common atomic organic elements in our universe, what kind of theories we know today about how life began on planet Earth. Remember lipid worlds, RNA world, even panspermia. We talked about habitability, which environments we can sustain life in. What is the point of having all these microorganisms if you can't do anything with them? Well, we actually do a lot with microorganisms, starting with food and drinks and medicine, and even all sorts of lifestyle things that we take out from microorganisms and help improve our life quality here. So today we're going to talk about detection. And I want to start with the biggest question of all is why search for life? And those who have been here before know that I always answer because they're there. But actually, what I want to say is that we want to search for life because we want to understand more about our universe. Some people are very pragmatic, and they show us that if we find other life forms, yes, we can use them, we understand them, and we can use them for the benefit of humankind. And everybody is basically, in our human nature, are really big explorers. So why search for life? Because it's there, and it's time that we find also life outside of planet Earth. What detection methods do we have? So basically, detection methods are stand upon the following three main items, I would say, or main ideas, main issues that we know today about. We're talking about various and different signals from life forms. If a flower blooms, it sends optical signals with its, shape and form and color. It also produces a lot of different pheromones and smells. And ultraviolet, it looks completely different. It has chlorophyll in it, which is a green pigment. It's an optical, or basically an electromagnetic signal that we can use. And so when we're looking at detection methods, we're really interested in finding the different signals that life forms can produce. And there's a huge variety of signals. It could be lipids in the membrane, it could be pigments, it could be other things as well. It could be perhaps sound even. Perhaps life can change the sound of a world, and we need to find out a detection method for that. I'm just speculating at the moment. But that's one point of a detection method. Look for something that we know a life form produced. Another thing for detection methods that they work on for life forms is to find accommodating environmental conditions. We know now what is the habitable zone or the Goldilocks area around a star, where in that area you will find liquid water if there are water molecules, and if there is water for a lengthy time, then we today believe life will evolve, life will emerge on a planet which has liquid water. But we also know, for instance, from hydrothermal vents, we know that if you have a rich nutrient and high temperature and high pressure, we can actually find magnificent ecological niches. So one thing that detection methods are trying to look out for, they're for environmental factors which will enable the creation of life. And then the last thing that detection methods really need, they need to be methods that are proven on planet Earth. No point in sending something out there which has not been tested and tested again and again on planet Earth to show that it can actually give you a clear and precise signal that this is a life form, or that this used to belong to a life form and not to something else. So we're looking for proven methods. And underneath, we're just sort of like looking for water, we're looking for temperature, different temperature, what are organic molecules. We're using these different things in order to get a sense of where life could be and how to detect them in the universe. So first, from a detection method, you're probably very, very familiar with this one. It's called, basically, we have satellites. And our satellites are very sophisticated nowadays, even though they get smaller and smaller, and we have nanosatellites and CubeSats and all that. Here you can see a very nice work done by NOAA, basically based on the NASA Sea-Wife sensor, which is a sea-viewing wide field of view sensor. This sensor was sent into space, and from 1997 till 2010, basically circumvented planet Earth and collected data regarding its chlorophyll. It was looking towards the ocean, looking at the sea surface. It's a very hard analysis to do on ground. Nevertheless, they were looking for the wavelength that would indicate the presence of chlorophyll, which is a pigment, an organic structure within the cells of not only plants, but also phytoplankton, which live in our oceans. And as you know, planet Earth majority is an ocean. It's not a land-based planet, it's an ocean-based planet. So things that are happening in the ocean are super important for the health of the planet and to understand how it works. And so a lot of chlorophyll is in phytoplankton, and you can see them blooming in different coasts around the world and in the ocean, and they do photosynthesis, and they really impact our CO2, our carbon dioxide cycles on the planet. So one thing that we could do generally is to use satellite to circumvent the planet that we are interested in and look for detection of signals of life forms, or in this case, chlorophyll. At the moment, we are not able to use the current stage telescope in order to see something like chlorophyll, especially when we are interested in chlorophyll, which is on the sea surface or on the land surface within a planet, not some in the atmosphere. And so we're looking for having perhaps better technologies in the future so we can send and look at the light coming from a very distant planet and understand whether it has some pigments. Chlorophyll is a molecule. It's pretty big molecule. It's definitely synthesis only by biological beings, and it will be a unique signal signaling that there is life form on a different planet. So satellites that are looking for signals. Another very successful mission in detecting or in the way of going to detect life forms is the Kepler mission. And the Kepler mission basically was looking for planets which will be in the habitable zone of their star. And between 2009 and 2013, this unique space telescope would basically look at one sector of the sky, looking at 100,000 stars for several years. And it will constantly make sure to understand whether there was a blip in that star light coming into this space telescope. And the idea was, and it turned out to be very successful, is that if you concentrate on one point of light constantly, and from time to time you see its light dimming, and it goes in certain cycles and repeats itself, then you can learn a lot from the dimming of this on what is this planet, what's its size, is it in the habitable zone around its star, what mass is it? And Kepler was so successful that today we have in our galaxy, we have 4,284 confirmed exoplanets, planets which are not necessarily the size of planet Earth. Actually, most of them are the size of Jupiter, which is 1,300 masses more than Earth. But they are nevertheless almost 4,000 confirmed exoplanets which circle their sun in a habitable zone, which means if there is water there, they will be in liquid form, and there's a good chance to find life forms there. The issue with Kepler, which had its main mission ended in 2013 and then had an extended mission till 2018, is that it was so good at detecting very far away exoplanets. We're talking about hundreds, even thousands of light years away. And as a result from that, they started working, and they just finished working and getting all the data from Tess. And Tess is NASA's Transiting Exoplanet Survey Satellite. And there's a very quick video here, which I will now press play on, that will explain this mission very nicely. Check it out.
Tess, the Transiting Exoplanet Survey Satellite, is NASA's newest planet hunter. The mission is operated for 2 years and has now mapped about 75% of the sky. This enormous area of coverage gives scientists unprecedented opportunities to search for new worlds beyond our solar system, as well as other cosmic phenomena. Tess completed its initial sky survey in July 2020. This giant mosaic is made from 26 sector images. Each sector is a 24 by 96 degree strip of sky monitored by Tess's 4 cameras for about a month. Here are some of the noteworthy discoveries from Tess's second year. Tess discovered a trio of planets around GJ 357, a red dwarf star just 31 light years away. The system's farthest planet, with about 6 times Earth's mass, is especially intriguing. It orbits in the outer part of the star's habitable zone. If this planet has an atmosphere thick enough to keep its surface warm, liquid water could exist there. Tess found another planetary trio around the star L98-59. The innermost world is the littlest found by Tess, and is 20% smaller than Earth. Later, scientists announced Tess had observed a star being torn apart by a black hole. Catching one of these tidal disruption events showcases the mission's ability to spot many kinds of short-lived phenomena, like stellar outbursts and supernovae, underscoring Tess's importance for studies beyond exoplanets. In early 2020, scientists announced Tess's first Earth-sized planet orbiting in its star's habitable zone. This world, called TOI 700d, is a little larger than Earth and probably rocky. Based on Tess and Spitzer data, researchers were even able to model potential weather systems. Analysis of the planet's atmosphere, if it has one, will have to await future space-based observatories like NASA's James Webb Space Telescope. Missions like Tess, and later Webb, help contribute to the field of astrobiology, the interdisciplinary research on the variables and conditions of distant worlds that could harbor life as we know it. Tess has found many systems very different from our own. TOI 1338 has a Neptune-sized planet orbiting a pair of stars. Kelt 9 has a fast-spinning star with hot poles and a cooler equator. Kelt 9b orbits over the poles, resulting in star-induced seasons that only last a few hours. Now that its primary mission is complete, Tess will return for another year-long survey. Many new planets, as well as more brief cataclysmic events, await astronomers as they scour the flood of Tess observations.
And that has been a beautiful presentation of the Tess project, which is led by the MIT Physics Department and has been sent to space aboard SpaceX Falcon 9 and has been managed together with Orbital ATK, Northrop Grumman. Now, an interesting thing happened since the days of Hubble telescope is that both Kepler and Tess are using their data and making it available for the public. So both Kepler and Tess have online archives and databases that you can just easily go into and start munching about and looking at the different data that they're coming. And if you're very talented, you can do your own exoplanet hunt. In fact, I do believe that one of the new things that Tess is coming out is that they realize that they need a lot of computing power in order to hunt all these exoplanets that they're intending to find with Tess. And with Tess, they're aiming to find, several thousands of exoplanets and dozens of Earth-specific, Earth-size exoplanets, and all within the range of 500 light years or even less than 200 light years. And they were really like the public help. So they have like an exoplanet hunting application, and everything is very, very approachable in NASA's website. And what's interesting also about this, this is Professor Sarah Seeger from the MIT Physics Department, and she's one of the leading experts today on the exoplanet and specifically on Tess. What you can see here is the results from the 2-year sky coverage map. And it's not, everything is not homogenic about this, but what we do see is that the minimum amount that the Tess was looking at a specific segment will be for a minimum of 27 days. And it's interesting to think whether in 27 days you will see that repetition of planets around the stars. Kepler was aiming at 100,000 stars direct several years. So it had them on constant surveillance for several years, while Tess has been shifting between these different section and sectors when it was moving around. And so it would be very interesting. The mission itself has finished, although as the video said, they're now starting to do again the southern hemisphere. But the data already exists in the archive. So it will be very interesting to see what other exoplanets Tess will be showing us. And as the video mentioned very briefly, there's a new telescope that is called the James Webb Telescope. And it's supposed to be like the next amazing level of Hubble telescope, only it's really behind schedules. And now with the corona, the next launch, it's already cost about 3.5 billion euros. It's going to be an amazing telescope. It will be so powerful that we will be able to look at distant stars and ascertain their atmospheric conditions, which is a huge thing for us because we don't know, aside from our solar system, we don't really know anything about atmospheres in other planets. And we're really interesting to know about the different, there's probably so many variations of what is an atmospheric condition of a planet. And in this condition, we know that now we have 20% oxygen. And that's actually a really big signal for us that life has evolved on this planet, okay? So cyanobacteria, all of a sudden you had photosynthesis, and only then did you have major amounts and concentrations of oxygen in the atmosphere. Unfortunately, we know microorganisms can also survive without oxygen. So finding oxygen in a different planet-sized Earth would be a very exciting finding, and I'm looking forward for this telescope, for this James Webb to be launched to the sky and to work properly. But already the data from Tess and Kepler gives us a good number about how many planets or exoplanets in the habitable zones we can find in our galaxy. So far in its 2 years, and again, it's just finished. It's still, there's a lot of data analysis to do, but still there's 1,835 candidate planets. And the beauty of Tess is that because it's looking at stars which are much closer to our planet, we can use ground-based telescope in order to confirm and to make sure that this is really an exoplanet revolving around the sun in its habitable zone. So I'm pretty excited about the next few years when it comes to Tess data. What else do we do with detection? What other detection methods do we have? So whether you know it or not, it's the age of the robotic oceanography coming aboard. This is a very nice slide by Oscar Schofield from the Rutgers University. They have an amazing lab, and they're using this little baby, which looks like a little torpedo, but it's not a torpedo. It's called a glider. And basically what these things know what to do is they know how to dive underneath and dive about 2 kilometers depth, 1 kilometer depth, and then they are able to record all the different physical parameters around them. So they're looking for pressure, they're looking for oxygen, saturation in the water, they're looking perhaps, I don't think they're looking for pH, but maybe they can, they're looking for temperature, they're looking for streams, they're looking for a lot of things, and they log. And once in a while, they go up to the surface and they transmit their data straight to the satellites, NOAA satellites, which then the satellites bounce back to the Rutgers University observation lab. And these gliders have completely changed the way we know how our oceans work. If we all the time, satellites are not good at looking in depth into the water, they're only good for the surface. So we had to rely a lot of, for instance, of submarine data, and a lot of that data is actually military data, so sometimes you don't have access to that data. So these gliders has revolutionized not only the science that we're doing, but also they were enabled students, high school students in the United States, to guide these gliders and understand how they find their way in the world and what type of oceans, what the difference is between the different oceans. And they have revolutionized their way on how understanding oceans. If we're interested in having a robotic mission to Europa, one of Jupiter's moons, then a glider is something that we know how to work with, and it's a fairly simple tool that knows how to look at the environmental conditions. And once again, I don't know if you've been here, but this is a beautiful video I took during my course at Woods Hole in 2013. And basically, there's in marine biology, we have now all these submersive. Some of them are crewed, some of them are autonomous and are used only, remotely controlled. But they have revolutionized our way of understanding what's happening in the depth of the ocean. And the depth of the ocean is not dead. It's not void of life. On the contrary, because there's so much underwater and there's tectonic activity and sometimes underwater volcanoes, there's actually a lot of things happening underneath that we never really imagined that they could happen. So it's really important to use these submarines, and Alvin was the first one in 1964 that has been used very successfully till today. Via Alvin, we discovered more than 300 new species at the bottom of the ocean, 24 new hydrothermal vents that we never knew were there. And it's an amazing time to be a marine biologist. So just let's listen for a moment for Dr. Julie Huber.
Nation with no ocean explorer, but they saw this amazing life on the seafloor. And in 1977, everybody still thought that basically the deep sea was dead. And if you listen to the original Alvin communications that were going on down in the submarine, one of the scientists in the sub called up on the radio and said, "I thought the deep sea was supposed to be dead. Why am I seeing clams?" I mean, it was that unexpected. And it was a few years later when Pauline Cavanaugh and others figured out that these tube-worm guts were packed with chemosynthetic bacteria.
So basically what we find is that submarines, even remotely operated, are a very useful thing. They come with high-definition cameras, so you can look. Their communication skills are pretty complex and are very easy to gain the data from them. Some of them have even like these mechanical arms that you can sample, you can put in a can, you can take the can, and some of them have room for a human crew. I actually met someone who was one of the first people to go in an Alvin together with her professor, and she didn't even know how to swim. But it was an amazing experience anyhow. So these type of sophisticated submarines are a pretty good tool in order to detecting things, because they pay attention to the environment, but they bring you proof, real proof that there are life forms in very hard areas to reach. So if we're going for a mid-summary about technology for life detection, so life detection goes for life detection technologies are based today on 3 different principles. One is there are different signals from life forms, so you can have different I'm everywhere today. Different methodologies in order to find these different signals and attenuate to them and find them exactly and know that they are coming from life forms. We're looking for accommodating environmental conditions. Perhaps if some place was had liquid water in the past, but now it doesn't, it will leave behind certain chemicals, certain minerals that we can check out and understand that there were habitable environmental conditions. All these different Kepler tests, all these places that are looking for the habitable zone around the star, that's looking for environmental conditions. We're not looking for life forms, but we are looking for the environmental conditions that are necessary from our point of view and from our understanding that life can be created and can emerge in these places. And we're looking for methods that have worked on planet Earth, that we can understand the data, that we can, if the beauty of Tess, for instance, is, like I said, that they can have also truth from ground telescopes inside planet Earth. So if we're looking for methods that can achieve several things, that would be the way to go, to find methodologies we can detect life, but also can cross-reference and understand what we're seeing. And that's the beauty of it. Also, what we talked about satellite, and satellites are a huge thing in Earth observation. We are using their capabilities in Earth observation, like I've shown you with the chlorophyll. But today we can actually know what's happening on the chlorophyll of a single leaf inside a meadow or in a farm, and we can detect a lot of things with gas composition and other things with satellite today. It's a big area that is getting a lot of funds. And what we're doing is, what we're aiming for is to have eventually satellites circling other planets perhaps, and giving us that data in that very high resolution way that we are used to on planet Earth. Telescope has always and will always be very reliable methodology for life, not necessarily for life detection, but at least to understand other solar system, other exoplanets, which might be Earth size or bigger or smaller. And as the telescope will get basically better, and James Webb is one example, but people are talking on putting telescopes on the moon, because the dark side of the moon is a very quiet place, and all the electromagnetic interruption that someone would ascertain on the moon, on the far side of the moon, would not be because of Earth, but because things that are coming from the universe without any interruption from planet Earth. So people are talking about that. Tess is an exoplanet hunter as well as Kepler, and there's a lot of data in the databases, and they're available for the public. So I urge you to try and find them and start, playing with those data and see what you come up with. We have gliders for marine oceanography and also submarine. So we're looking for, these are methods that work with liquid water and are looking for environmental conditions. And in the case of Alvin and some of the more sophisticated submarines that we get today, they can be actually crewed or autonomous, and they can actually give us bona fide facts or proof, actually, that there are life where they're going without any hesitation whatsoever. So moving forward, there's something which is called laser-induced breakdown spectroscopy, LIBS. And I need you to know this because this has been one of the best methodologies for NASA to send on space mission, and that's why you should pay attention. This method has been proven many times on Mars and also on planet Earth. We use it you point a laser, the laser produced plasma, and then you can point this laser, of course, you can point it at solids, at liquid, at gas, at rocks, at ice, at soil, at the human tissue, whatever you want to do. You can charge this laser, you can produce the plasma, and what happens is that as the plasma cools down, there is this process in which the plasma has been absorbed. If you have a molecule which has certain atoms in it, then the atoms, due to their number of electrons, can absorb certain wavelengths. And then they absorb what they can from the plasma. And the remaining wavelengths are being transmitted out. This pattern that is being transmitted out is very characteristic. Water has a very characteristic wavelength spectrum. If you pounce it with a laser produced, if you know exactly what is the laser, what is the intensity, how long have you pointed to the water, you will get a very certain emission. And so what happens today is that there's big libraries actually of spectral peaks of this method, of this spectral peaks. And basically each element in the periodic table is associated with a unique LIBS spectral peaks. And the way it looks, it's somewhat in what you see here on the screen, is that in the middle of the screen, you see how a molecule, how we build it in a 3D or actually 2D dimension, and this is called a pantaerythrol. It's not a very complex molecule, but once you use the right plasma for it, once you use this technique, instead of seeing it as a molecule, you're going to see an output which basically has a lot of peaks in it and different wavelengths. And what we know today is that we see these peaks on this output, and we know exactly what each peak means. This means a connection between a carbon and a hydrogen. The other peak is between nitrogen and a hydrogen. And when we accumulate it and we look on the entire set of these peaks, we know exactly, probably at high probability, what molecule it is. And this is important because this has been one of the most used methods on the rovers on Mars. They would point a laser at a rock, get this plasma coming out. They would detect what kind of peaks are coming out, and they would send out, okay, we found this and this mineral. We found this, carbonate base, whatever. And it's amazing that this technique is working so beautifully, and it's been used many, many times on NASA's missions and also on planet Earth. When we look at other things that are more interested for microbiologists, is that there are a few things that need to be taken care of. First of all, you have to realize that there could be hundreds of thousands of species of bacteria in 1 gram of soil, right? And what happens is that during the 1990s, microbiologists started to realize that when they take 1 gram of soil and they put it on a petri dish and they grow it in the lab, they don't see, thousands and thousands of species. They would see between 5 to 10, maybe just one. So there was something which is called the plate, the big count plate anomaly, which meant we know this is a very rich soil, but we don't see it in the lab. So what's happening to the rest of them? Why are they not growing? And what happened is that as we progressed into molecular biology, and we started to look straight at the DNA of things instead of trying to grow them in the lab, we were able to ascertain that gap and understand exactly how much species of bacteria are in 1 gram of soil. So in generally, there could be billions of bacteria. Yeah, 1 with 9 zeros, bacteria in 1 gram of soil. And it would be interesting perhaps to know, but the species in 1 milliliter of seawater is a lot less, like maybe 1 million. So what happens is that people are using a lot of microscopy in order to detect life forms.
Hello, Reut, are you there?
And how it works on the right side. There's
sorry,
a nice
we
diagram.
lost you for
yes, I'm here.
Yeah, we lost you for like 2 seconds. Also, if you want to just drink some water, that would be fine if it's troubling you.
Yeah, I don't have a lot of stuff here, so it's gonna be fine.
Okay, good.
Let me know when I can go again.
Yeah, no, it's on. We just, I think the internet had an issue just for like 2 seconds. It's back, no problem.
Okay.
Yeah.
Great, thank you. So we have light microscopy, which is basically you take some source of light, you put it through through different types of lens, through the microscope, then you can view the image, and the lens basically blow up the image that you look at, and we have very high-end microscopes today. Fluorescence microscope is what produced these beautiful pictures that you see now by me. Basically what happens is that you don't just put white light on your table of examination. You're using fluorescence. You're using fluorescence light at specific wavelengths in order to excite the different things that are inside the microorganism you're looking for. You're looking at microorganisms which has I forgot the name for it for a minute, which has different types of pigments that are able to, on one hand, to absorb this fluorescence and emit emit light at a different wavelength. And when you're using an epifluorescence microscope, basically you see the images of the different microorganism, but you see them glowing if they have this pigment. If they don't have the different pigments, they won't glow. So what's interesting here today is that you can attach fluorescence markers to DNA, to RNA, to membranes. And basically what you see here, for instance, on the left side, you see this cone shape with 2 green lights and 1 blue in the middle. That's because that's a diatom. It's a marine phytoplankton. And basically what's glowing up in there is a certain marker, which I added fluorescence dye to it. And that marker attaches only to the DNA and to the nucleic place of this diatom. So what's lighting up over there, that's where the genetic code. So when you use this technique, you can localize different organelles. You can localize where the genetic code is being assembled, where it's being kept. Perhaps if you want to work on the ribosome, then you just you put your markers over there. And so you are able, on the right side, you see a green and you see a red. The red is actually how do you see chlorophyll under the microscope? And it's red, and it's very lengthy, and it's red. And you see all these other green stuff around it, because the green stuff, again, it's a different type of bacteria, which is sort of like having a ball and having a good time on what it looks like a red, but it's actually a chlorophyll-producing bacteria, most probably a filamentous cyanobacteria. So when we're using the microscope, and today there is a lot of microscopical, almost like an art form, the things that we can do today with microscopes. We can interlay, we can overlay the different images, we can understand what happens in time sequences, we can do a 3D images. The only thing that it takes really long time to prepare one image or one specimen to make this happen. And actually, when you walk to the different level, to a completely different level, which is the electronic microscope, with the electronic microscope, it takes between 4 to 7 days to prepare your samples, because it has to be completely dry. It has to be very meticulously done. It has to be coated with gold, because electron microscopy doesn't work on the optical level at all. It works already on the atomic level. It's a completely different setup. So how are we going to detect life forms on Mars? Are we going to send in a microscope? So one of the things that is happening today, I don't know if this is the next one. Oh, sorry. So one of the things that we are able to do, but only in liquid environment, is that we're able to use a cytosub, which is a submersible microscopy. It's a certain type of a submersible equipment that alongside can really count and can take images of the microscopic organisms which are floating in the water. And that it works for several hours. It's a very delicate submersible equipment, but it does give you this sort of like ongoing, for several hours, you just put it in the ocean, hopefully a calm ocean, and then you can see what kind of microorganisms are able to live there. So if we have an ocean on Europa, if we have an ocean on Enceladus, maybe one of the things we wanted to consider is a cytosub. Or maybe we don't want to use it because it's too delicate, perhaps. But these things are able to find exactly life forms and microorganisms life forms in different environments. So I was telling you that.
PCR is a powerful.
So I was telling you about this molecular method that started to look on different things, and it looked on soil, and it looked on plants, and it looked on even on different rocks. And we found out, even in seawater, which have a very low quantity of bacteria, we found out that actually there is a lot of copies of DNA floating around in bacteria and viruses and everything. So actually everything is full of life. And the reason we were able to do it is because of this polymerase chain reaction process. And the guy who won the Nobel Prize, even though there were several groups perfecting it, is called Kary Mullis, and he won the Nobel Prize in '93 for the PCR. And he was able to alternate it and add the Thermus aquaticus DNA polymerase to the process. So this is a very nice video explaining what PCR is. For those of you who don't know, it's very nice. And it's made by a company called Mini PCR, which was the first company to actually produce a very tiny machine that is able to do this process. And they also the very first company that actually sent it to space, to the International Space Station. And a student in 2016 was able to perform her experiment on the International Space Station using their device. So this is what is PCR and why is it so great? Here we go. Oops, sorry.
PCR is a powerful technique that allows us to make billions of copies of specific pieces of DNA. The first step in PCR is to open up the two strands of DNA to expose the genetic information encoded by the nitrogen bases, A, C, T, and G. In the next step, custom-made DNA molecules help us find the beginning and end of the DNA piece of interest among the billions of letters in the genome. These genetic bookmarks are called primers. The third and final step is to fill in the genetic information between the primers. An enzyme called DNA polymerase adds the building blocks of DNA called nucleotides to create an identical DNA strand. DNA polymerase works in our own cells to replicate our own DNA each time it divides. The first step in PCR is known as denaturation and is enabled by high heat. The second step is called annealing, and different primers have different and very specific annealing temperatures, typically between 50 and 60 degrees Celsius. The third step is called extension. DNA polymerase used in PCR works best at 72 degrees Celsius. In these three steps, we've taken our original DNA molecule and obtained an identical copy of a very specific fragment. If we repeat these three steps one more time, we'll go from 2 copies to 4. Repeat it again, and we'll obtain 8 copies. And again, and we'll get 16, and 32, and 64, 128. Well, you get the idea. DNA amplification by PCR is an exponential process. After 30 copy cycles, we will have 2 to the 30, or 1 billion identical DNA copies. This is enough DNA, for example, to detect a pathogen in blood, such as HIV or Ebola, or to amplify trace amounts of DNA found at a crime scene, or to enable any of the other powerful applications of PCR.
So I hope that was clear enough. What I mean with this is that PCR is now being done every year, perhaps 1,000,000 times, if not billions of times, on this planet. It's a proven method to find DNA, to find RNA in a lot of different types of origins, whether it's a rock, ice, soil, solid, liquid, and so on and so forth. However, it is a very sensitive methodology, and it is, based on our way of life, our nucleotide, our DNA, which has evolved on this planet for 4.5 billion years. Has DNA evolved exactly the same on other planets? We don't know. So while this is a great method to detect DNA and RNA on planet Earth, we're not sure this is a great method to detect anything else elsewhere in the solar system. But it's worth to note that this is one of the main tools we use today around the world to detect DNA, RNA, and basically understand our entire world around us. So let's take a case study and think about it for a moment, okay? Let's take this. This is ALH 24001. ALH stands for Allen Hills. It's an area in Antarctica. It's a meteorite. It was found in 1984, okay, in Antarctica, and it weighed almost 2 kilograms. It's, according to its chemical compositions and isotopic and radioactive isotopic analysis, it's about 4 billion years old. It came from Mars, and it came from Mars about 13,000 years ago to planet Earth, but it comes from a very old region in Mars. And in 1996, a scientist called David McKay published in Science this photo, this black and white photo that you see here. And you have to wonder, is that thing like a fossilized, perhaps, of some sort of bacteria? And it's interesting. So if you know of all the different detection methods that I've just spoken about, you might suggest a few more things to do before you realize just because it looks like a bacteria, not necessarily means it is a bacteria. We need to check other things as well. Another thing to pay attention to is that whether this section was taken from inside the meteorite or from the outside. And was the meteorite completely exposed to the atmosphere of Earth? then maybe something has landed on it like 13,000 years ago, and what you're looking at is not Martian at all. It's basically some germ that landed when the wind swept in Antarctica. So now it's quite clear that this is not a Martian fossil, but at the time it caused so much discussion and debate that even Bill Clinton, as the president, had a special speech to the country addressing it and then saying maybe we found life on Mars and this is the evidence for it. You have to realize that when we to detect life, we really need a whole battery of tests coming from different areas of science, whether it's checking for physical or chemical analysis, looking at the history and the geological history of the area, and then looking for biological molecules. Usually they have a lot of carbons, they have a lot of hydrogen, they have other, whether it's phosphate or nitrogen or other elements integrated into that molecule. So it's a big question how to find life elsewhere. It has not been done yet anywhere else other than planet Earth and its extreme environments. So let's, again, another case study. Let's take a look about what's new about this NASA Perseverance rover, which is going to Mars and supposed to land in February 2021. So basically, Perseverance is a big vehicle. It's going to work somewhat like Curiosity. It's going to land in a completely new area, which is called Jezero Crater. And it's an amazing crater because we know for sure it had water 3.5 billion years ago. And if it had water, there might be other stuff living there at that time. Whether they left something, we don't know. But what is the equipment that has been sent with Perseverance in order to detect these life forms? So what we now, when we look at the NASA website and we look at all the different details, and by the way, there are magnificent images and 3D and videos, and you're welcome to look at mars.nasa.gov for all the information that your heart desires. We see that it comes with a total of 23 cameras, and 7 are science cameras. The rest are for engineering and making sure that it doesn't trip too much or that everything is okay. The other new items for Perseverance is that it has new wheels, it has a new software. It's going to collect rock cores. So there's an extended new robotic arm which has been tested on planet Earth, I don't know how many times, but it's supposed to drill a hole, collect whatever in that hole, and then put it in a special container. That's why it has a large hand, or turret, as they call it. So what are the 7 science cameras? How are they going to find out if there's life on Mars? So first of all, there's a camera called Mastcam Zim, which is a pair of cameras, and they take color images and video, 3D dimensional stereo images, basically looking at something and saying whether life perhaps have been there. So if they would find a really, really big fossil, then those cameras might catch it, and you will look at it and say, okay, we have a big fossil here. Another option, which is more problematic, is what happens if they find that the layers of there are different layers of rock, right? And those different layers perhaps maybe were constructed by stromatolites. Can I tell that only from looking at it? I'm not so sure. And you need more and much more work done in order to ascertain that those were once a life form in Jezero Crater. It has a Supercam. Now, the Supercam fires the lasers, creates a plasma, and an onboard spectrograph records the spectrum of the plasma, which reveals the composition of the material. So this is exactly LIBS, and LIBS have been done in other rovers as well, and it's a very successful and reliable method of checking the mineral composition. So if we find organic molecules, really depends which type of organic molecules, by the way. Not every organic molecule has been created by a life form. Some of them have been created in a moist environment. But if it does find something that looks, wow, that gives you exactly the spectra of a chlorophyll, then wow, we're over the moon. Or of a lipid, which is part of the membrane of the cell, wow, we're over the moon. But if it's something else, we're not so sure. We have to look what kind of organic molecules have been left there. So that's the Supercam. And the Pixel L uses an X-ray fluorescence to identify chemical elements in target spot. Basically, it's very similar in that way in the same, I would say, same principle and how the Supercam with the LIBS work. But this one, it's using X-ray fluorescence in order to really shoot out the electrons, get a big spectrum out of it, and take a look at that spectrum and see what that can be. Sherlock and it's Sherlock is on. If you see in the picture, Sherlock is mounted together with Watson on the hand or the turret of Perseverance. So Sherlock's main tools are a spectrometer and a laser, but it also uses a macro camera to take extreme close-up of the area that are studied. So we have 3 cameras, which are basically some sort of a laser or something else that is point out in order to make an excitation of the whatever you're sampling, of your sample that you're looking at, and looking at the spectrum out of it. So we're having a lot of spectrometers because that's the proven method to work on Mars. But do we have any other methods going around? So we have Watson to capture the larger context images for the very detailed information that Sherlock collects on the Martian mineral targets. So Sherlock and the Supercam and Pixel are working on tiny areas, even less than millimeters, on tiny areas. And then there is a scientist back home who's looking at this and says, wait a second, but is it relevant for the geology of the area? What type of rock was it looking at? What is the shape of the rock? They're trying by using both the camera and both the spectrometer to get the context right of what they're looking at and whether it makes sense, because that's a really, really interesting thing to do. But so far, we haven't heard anything about finding life forms. We're just finding things which are helping us to understand the minerals, the composition of the molecules. And the last camera that is on the Mars 2020, now known as Perseverance, is a cache cam. So this is a new camera, and its sole point is to look down at the top of the sample cache. So basically, after they are taking what they're drilled and they're putting it into a tube, they have a camera above the tube to see how it looks, how the things are inserted inside. And that's also to understand if everything is working okay and whether they can close this container appropriately for sealing and caching. And then they're planning on a sample return mission, I don't know when, maybe in 2024, to go get those containers and bring them back to planet Earth. So that was one. There were 2 case studies that we were talking about. One was about the meteorite from Mars that landed in Antarctica, and the other one is about Perseverance. And what I'm trying to show you here is detecting life is a pretty hard and complex area to do. And we have different types of methodologies, which I think I only covered maybe just the main ones, from the really big ones, looking at exoplanets, to the small ones, looking at molecular, DNA, and things like that. I can't cover everything in this introduction. But you have to realize that it's really hard to detect life anywhere. On planet Earth, we've been doing it for several decades, and we're getting better at it. But once we step out of this beautiful ball of life, we're in deep, deep trouble. And we need to use as many tools and as many methodologies in order to understand what is really a life form and have we detected it, whether it's on Venus, whether it's on Mars. And we need to understand to employ as many methodologies and techniques as possible. And if you stay with me and you'll go with me to the next session that we have next week, and then we're going to talk about really life in the universe and what is the Drake equation and how the knowledge that we gained from Kepler and Tess, how does that intensify our way of looking at life elsewhere. And you're most welcome to bring your own materials as well. I'd be happy to talk about it then. It's a big discussion about life in the universe. So thank you very much. And I would like to recommend these 4 amazing books for you. Astrobiology, which is a very short introduction by Professor David C. Catling. It's an amazing book. It's super short, and it has everything you ever wanted to know about astrobiology. It's awesome. I really, really like the I Spy books. I think every explorer at heart would love these books. There's a lot of riddles in it. You need to find hidden things. It's exactly like a scientist work. We are looking for stuff. We're trying to find them, and we're getting a lot of riddles on the way. So I really like the I Spy book. They make fun out of my job. Alien Ocean is a really new book. I just started reading it. I got it in the mail yesterday. It's an amazing book by Kevin Hunt. It's about the search for life in the depth of space. And we're talking about the different oceans in planet Earth and the different oceans elsewhere in the solar system and how we can find life, what we should be going to, how it's going to feel for us to do those missions. And a very classical book for everybody who loves Mars and wants to know more about it. The Case for Mars, The Plan to Settle the Red Planet and Why We Must is the latest edition by Robert Zubrin, who was an NASA engineer, and now he's the president of the Mars Society. And he's an avid speaker for the fact that we need to go to Mars as humans. We need to settle it. We can do it. He has a lot of engineering solutions for it. And if you're looking to read one of the greatest minds on how to deal with Mars, I highly recommend this book. Thank you very, very much.
Thank you so much, Reut. And yes, it's question time now. And I really love that you enjoyed about the rover that we sent to the Mars right now, because I think everyone's been, interested into it. And the way you explained all the cameras and how the equipment up there works, I think everybody, really enjoyed it. So yeah, let's go to the questions. What kind of pigments does organisms which live at very depths of ocean near geothermal vents have, and how does the evolution of pigments has helped organisms? Did you get that?
Okay. Yes.
Yeah.
That's a great question. Okay, it's actually a couple of questions, right? So what happens with pigments underneath of the ocean? You don't find a lot of pigments, mainly because pigments work well when you have light hit them. And when you don't have the light hitting them, you can't do a lot of work with them. But we do find pigments there, and we think that the organism down there, some of them don't really need eyes, so they wouldn't use colors, but some of them might maybe use the color in order to do something thermodynamically, or use it in a different wavelength, perhaps UV. I don't know. There's not a lot of research done. It's a great question. But we find, for example, in caves, we find albino crabs, and we find albino fish, and we find albino a lot of things, because there was no point in the dark to produce any kind of pigment. It's sort of like a waste of energy. So that's a great question. We find some pigments on the depth of the ocean, but not as rich as you would see, for instance, in 6 meter underneath the ocean, when you still have the photic zone for the light to come in. What was the second question in that question? Something about evolution?
Yeah. How does the evolution of pigments has helped the organisms?
So, pigments do a lot of things. Actually, it's a big family of molecules. Some pigments are there to do communication with other species, if we're talking, big animals and things like that. But other pigments, when you look at microorganisms, mostly the pigments are there either to do photosynthesis, and photosynthesis on different wavelengths, by the way. We have chlorophyll, alpha, beta, gamma, and delta. We have different types of chlorophyll, which are adapted a little bit to different types of wavelengths to do the photosynthesis. So it depends where this organism lives, how deep it is. Maybe it's in an area that for some reason gets a lot of red light, and it will develop the correct or the most suitable photosynthetic apparatus for it. And another thing is that they use the pigments. For instance, chitin is a black pigment that you can see in a lot of places in desert areas or other places which have direct sunlight and direct UV. So this pigment, what it does is that it absorbs this UV, and it excites, and it emits some electrons. But by collecting the sunlight energy, it works as a shield. This pigment is the one that absorbs most of the energy that arrives because of this, energetic UV or energetic solar energy, and it provides a shield from other things harming different other structures or other molecules within the cell. So that's a type of protection that the pigment. So we have for communication, we have for protection, we have for harvesting photosynthesis. So I'm guessing it really depends. Maybe there are pigments which do all three. So I'm thinking the more you need them, the more you will have them. Basically, that's about the evolution of these things, and it's a complex metabolic structure. By the way, it has a lot of stages until one pigment is formed, and some places have thousands of pigments within one cell, and others have none. So it's a big thing. A pigment is a huge topic. So thank you for it.
Okay, got it. So this question was by Adarsh Kumar, and he's asking another one. Can you explain a bit more about the KAI map of Tess 2 year sky survey? Like how was it made, and what its significance?
KAI 2 map. Have I got it correctly?
Map. Yeah, of Tess 2.
Yes.
Yeah.
Of Tess 2?
2 years
sky
sky survey.
Kepler.
No.
Yeah.
Okay.
I thought it was Kepler.
Maybe.
Yeah, I have to look into that. I don't know the answer offhand.
Okay, fine. That's okay. We have another question by Miss Nirgude. Sorry, just a second. So many questions today. Um, does the solid water contain any life samples? If not, then why?
Okay, so the solid water, you mean
Yeah,
like ice? So
yeah.
basically, yes. So what I want to say is that they're great. So first of all, have you heard of permafrost? Permafrost is a thermodynamic process. Basically means frozen ground, but during certain times of the year, only the upper layer sort of like gets a little bit liquidized and then frozen again. So there's this thermodynamic process going on with the permafrost. So what we find is that in the permafrost, which is basically frozen ground, we have a lot of bacteria that were trapped there for literally thousands of years. So there is life there.
Mm-hmm.
In ice, and I used to work on ice cores from Antarctica a little bit, it's very hard, but you can find DNA in ice. So I would say there is life forms there, but it's very hard. It takes many days. You have to process hundreds of samples, and then you have to be very particular. So yes, in solid water, you can find signs of life. I don't know about whether it's now active or not. Does that make sense?
Yeah, it absolutely does. I was wondering while you were explaining this, then why don't we send any rovers or missions to the ice polar caps of Mars then?
That's a great question.
Yeah, it would be interesting. Maybe you can tell this to your NASA friends. Okay, moving on. She has another question. So she's asking that you mentioned about the soil which has bacteria. So she wants to know if there is any specific soil where which can contain such bacteria. Are there types of soil which doesn't and types of soil which does?
Yes. So there are many types of soils. If you ever want to look at the different types of soil, just go to your local, I would say, agriculture ministry or agriculture center, research center, and look for the different types of soil from your country. In Israel, there's at least 200 types of soil. In the United States, maybe through their websites, which are sometimes more accessible, you can find even more types of soil. So the issue is, it's a big issue. Basically, from what I know, if you take sand, quartz sand, let's say from the Sahara, you will have a very hard time getting DNA out of it. And there's not a lot of DNA in that. There's not a lot of bacteria in sand, which is a type of soil in my opinion. So that area, for instance, won't have a lot of bacteria. But when you look, for instance, on soil of agriculture, soil which is around human populations, forest soil, there's so many different types of soil around the world, whether it's a meadow, whether it's a mountain, whether it's everything, you will find bacteria in that soil. It's a very prevalent thing of planet Earth. We have a lot of life here.
Yeah, and maybe the soil that is deep inside of the sea, maybe there as well there is great chance, right, of finding some DNA.
Yes. So that would be a very protected area. Basically, any oil, I would argue that any oil reservoir would be filled with DNA, but it will take a long time to produce that DNA in a clean fashion so we can work on it.
Yeah.
But yeah, definitely sometimes it depends how deep it is. It depends how many years it has been laying there. There have been many research vessels which were doing exactly that. They would take a core from the depth, from the soil of the ocean, and will extract that huge core. And from that huge core, in the same way that you look at, let's say, bubble gases from Antarctic ice cores, you would look again at all these different layers, and you will know a lot about the geological issues that happen within the ocean. And you will find the prevalence of different species. And from the beginning till the end of the core, you might find a complete shift in the ecological way. And that's because our planet has been very dynamic through eons. So one place would be, without any oxygen, and then all of a sudden oxygen will enter, and things that would have been in the photic area and would enjoy oxygen eventually will float down and be buried in the sediment, and you will find them. So it's a very dynamic area of research to look at these cores. Yes.
Yeah, so I think it makes sense then that Perseverance rover was sent to the area where the water did exist before. If there was sea or something like that, the life there would have been, preserved very, very well. And talking about the seas, it would be completely different case for the Dead Sea, right?
Yeah, so the Dead Sea has a lot of bacteria, has viruses that are related to salinity, and has little shrimps which are related to salinity and are able to survive. There's even in the area which are just a little bit less salty, there is a type of fish that is able to survive there.
Oh.
So it's not really dead.
Yeah, I got some of the sea salt for myself from the Dead Sea. I just picked it up from the Dead Sea and got it home. I've preserved it a little bit. Okay, so moving on. Question from Sanika J. How do we know that it comes from the Mars? I think she's talking about the meteorite that you showed. You were talking.
Yes.
Yeah.
Yeah, LAH. So there's Murchison meteorites, and basically what we know about them is that we know the composition of the rocks is very different than the composition of the rocks from planet Earth. And I don't remember the exact composition at the moment. But basically, when you look at the composition of the rocks, and also when you do an radioactive isotopic analysis to see what kind of elements and how much, how much decay the atoms were there from the different elements that were in the rock, then we now know after we've sampled Mars that they come from Mars. For a while we knew that they probably didn't come from Earth, but we weren't sure that they came from Mars. But then we sampled rocks from Mars, and we know for sure that that's the issue. So it's about mineral composition and radioactive isotope.
Okay, we have another question by Adarsh Kumar. In the survey question, I was referring to the Kepler data. So can you look into that again if you can? Maybe you can just send me the answer. I would put it up on as our Instagram story, or just post it in the comments or description over here later on, if you can just send me the answer. Okay, so guys, there are two announcements that I want to do right now. One is that Space Week is starting from today. So every year from 4th of October until 10th of October, we celebrate the World Space Week. And we are lucky today to have Reut talking to us about astronomy. She also mentioned some important work done by satellites, which is the topic of the World Space Week this year. So thank you so much, Reut, for this session. And
My
for the
pleasure.
next session, we all are extremely excited about the future of life and the aliens and stuff like that. So yeah, going to the announcements. The first announcement is that we are going to come up with a DIY satellite making competition. So if you make your own satellite, take an image and send it to us, and we'll post it on our Instagram channel, and there's going to be exciting gifts for everybody who participates into this. The details will be released tomorrow on our Instagram channel. You can find us at AstronEra. Also on our Facebook page, we will post it. And the second and the best announcement for the day is that after the sessions with Reut are done, we are going to have more sessions about the high energy universe by my friend from Turkey, whose name is Ozgur, and he is going to join us from 18th of October. So I hope you guys are looking forward to his sessions as well. And we have one more session to go with Reut, which is going to be, I'm sure, like she has reserved the best for the last. So we are all looking forward to that. And until then, clear skies.
Okay.
Yeah, I'm just still waiting for this to finish so that I can stop streaming. And